Hello, I'm Chia-Chun Hou. Today I'm going to talk about this project. This project is part of the biotechnology core technique class. In this class, I already put GUS DNA in E. coli. So now I want to get the GUS RNA from E. coli. The topic is use logistic regression to explain how IPTG concentration related to GUS RNA. First, I put GUS DNA in the downstream of leg operon. And in our system, we got a very strong repressor, so I cannot get any GUS RNA. Then I add IPTG, the inducer, to change the conformation of repressor, and then I can get the GUS RNA. The goal of our experiments is to find the optimal IPTG concentration. That's what gets the maximum GUS RNA. The method we use is first, we incubate bacteria for several hours, and in different conditions, we add different concentration of IPTG to see which one would get the maximum GUS RNA. And here is the 8 condition we use, and in each condition we do 2 replicates. The concentration of IPTG is between 0 millimolar to 1 millimolar. The strain of E. coli we use is PQG11 and 15. This strain contains GUS DNA and the repressor of leg operon. And the first result is narrow drop, and we can see the RNA concentration in different conditions. Why are we doing this? Because in the next experiments, electron fluorescence, we want to add the same RNA concentration in each sample and to compare the GUS RNA level. Here is the concentration of IPTG. And this 2 band is ribosomal RNA in E. coli. At the bottom is tRNA. And GUS RNA is between the 2 ribosomal RNA, so you cannot so clear in here. And you can see these 2 bands seems more brighter than others because I think there are some bias in narrow drop. At the right, you can see the northern hybridization results. Here is the GUS protein, GUS RNA. And we can see that when the IPTG concentration get bigger, the band seems more darker. The northern hybridization result is more accuracy than electron fluorescence. And this is our second replicate. As you can see, here is the different concentration of IPTG and 2 ribosomal RNA, tRNA, and not so clear GUS RNA. But in northern hybridization, you can see the GUS RNA in here. And there are the similar pattern is that it seems more darker in bigger IPTG concentration. And we want to calculate how bright are these bands. So we use IMAGE to calculate that. First, we calculate the area under the curve. You can see how bright each band is. And in northern hybridization, we do the same thing. But when we photo this picture, it seems a shift in there because when we photo this photograph, the light is brighter in one side, and I try my best to reduce the shift bias. Here is the results. First is the electron fluorescence, and next is the northern hybridization. And as you can see, this is the IPTG concentration, and the intensity of each band, and the intensity is the amount of GUS RNA. And you can see there is a pattern that's when the concentration get more bigger, the intensity get more bigger too. Next, we do the same thing in the next replicates, and we also calculate the area under the curve in electron fluorescence and northern hybridization. And we also do the same thing. You can see a similar pattern is that when the IPTG concentration get bigger, the intensity also get bigger too. So I want to fit a model for my experiments. First, I draw box plots, and you can see that actually when the IPTG concentration get bigger, the intensity get bigger too. But because there are some bias in each replicate, so I use the mean intensity to draw this plot. This is the IPTG concentration, and this is the mean intensity by using each replicate. And I realized that logistic regression is very good to fix, is a very good model to fix my experiments. And so I think that's the 0.5 millimolar IPTG concentration is the optimal concentration to get the great intensity of GUS RNA. You can see in this graph that when IPTG concentration get bigger, the intensity also get bigger. But when the IPTG concentration is bigger enough, the intensity seems the same. So I think 0.5 millimolar is the best concentration. And in this Nature Genetic paper, you can see the similar results from our experiments. This is the IPTG concentration, and this is the mRNA per cell. Is similar to our results. And in this experiment, I want to thank my partner Patient Lin and my teacher and professor Zhiren Chen. And I spent a night to think which model to fit my experiments. And finally, I found out logistic regression is a good model to fit it. Thank you.